Let's talk a little bit now about clinical trials. And this is something you probably heard a lot about as we've been following the SARS-CoV-2 vaccine development. And I want to break it down a little bit for you. So clinical trials in general have many different phases. There's a preclinical trial phase, and then there's the actual clinical trial, which has three phases: phase 1, phase 2, and phase 3. In the preclinical trial phase, what's generally done is studies in non-humans. So you're looking at mammals that are primates or rodents, and you're using them as a study system for the vaccine itself. You're making sure that the vaccine works in those systems and that they were safe. And assuming they have really good data on both the efficacy and the safety, now you can take that candidate and bring it into a clinical trial. In the actual clinical trial, then, what you're looking at is data from actual humans. In the phase 1 trial, what you're looking at is just safety. It's actually a very small study, usually involving 30 to 50 participants. And what you're looking for here is just that there isn't anything unexpected that happens when you use this vaccine candidate in humans. You want to make sure that it's well tolerated, that you don't have any crazy side effects that you didn't see in your mammalian systems. Now, assuming you passed your phase 1, you go into phase 2. In phase 2, now what you're going to look at is actually an expanded safety and dosing. So now you're going to take your vaccine candidate and you're going to actually try a bunch of different doses. And in fact, you might use doses that are up to 10-fold in difference. And you're going to try different schedules. So you might try 1 dose, 2 doses, 3 doses. You might try different periods of time between those doses. And you're going to then have many different cohorts in your phase 2 that are actually receiving different schedules and different doses. In phase 2, you are also looking at safety. So the thing that's being recorded and studied over time is what are the side effects that all of these individuals are experiencing, and is this vaccine being well tolerated at all the different doses that you've tried. Based off of your phase 2, you're going to take your most promising doses and schedules and bring that into your phase 3. So you'll probably rule out some doses and schedules from your phase 2 that you don't want to use in your phase 3, and you only take your most promising schedules and doses and bring that into your phase 3. Now, in a phase 3 clinical trial, you're actually now looking at efficacy. This is the first time you're actually evaluating how well does the vaccine actually work to prevent the disease that you're trying to prevent. In a phase 3 clinical trial, this is a very, very large study. Now you're going to look at doing probably tens of thousands of individuals being recruited into your study. In a phase 3 clinical trial, you're looking at efficacy. So what exactly is efficacy? You've probably heard this word used a lot. And one of the things that you probably also have heard is the word effectiveness. And they're used somewhat interchangeably, but they're actually different concepts. Efficacy is how well does your vaccine work in an ideal world conditions. Effectiveness is how well does your vaccine work in real world conditions. The reason is because in a phase 3 clinical trial, you're probably recruiting mostly healthy individuals. You don't have people who are otherwise very sick. You don't have people who are immunocompromised. You don't have people who have a ton of comorbidities. And the other thing is you have all the vaccine being administered by very well-trained staff who are there to make sure that every time they give a vaccine, they do it in a very controlled way to minimize side effects. In the real world, you're going to have participants that have all sorts of other potential complications. They might have other comorbidities, they might be immunocompromised, they might be otherwise unhealthy. And the people who are actually administering the vaccine are going to be all sorts of people across the world. You might get vaccinated in a pharmacy, you could get vaccinated in a drive-through clinic, you might get vaccinated at your doctor's office. So there's going to be a lot more variation in terms of who's actually administering the vaccine as well. What that means then is that your efficacy data is usually your best case scenario. Your efficacy numbers are going to be in the best case scenario, how well do you think you can protect against infection? And effectiveness is going to be somewhat, hopefully as high as efficacy, but could be somewhat lower than the efficacy, because in the real world where you have real full populations as well as real world conditions for giving the vaccine, you might see effectiveness go down. You might also see that the side effects might be slightly worse in the real world than what you saw in your trial. Now, for the SARS-CoV-2 vaccine candidates that have been coming through so far, we've seen a lot of efficacy data where they're looking at symptomatic COVID-19 as the endpoint. So they're actually looking at, do you end up with a symptomatic case of COVID-19 if you get the vaccine candidate versus if you get a placebo? In a phase 3 trial, you actually have many different cohorts that are all going to receive either the experimental vaccine or receive a placebo. What's very important in phase 3 is that everything else is the same across these two cohorts. So you want to make sure that there are equal number of individuals in both cohorts. You want to make sure those individuals are relatively similar. They have the same demographics. They all have the same age distribution. They all have the same gender and ethnicity distribution, geography, etc. So you want to make sure that everything else is relatively controlled. And the only thing you're testing then is, did you get an experimental vaccine or did you get the placebo? What's very important is during the course of conducting a phase 3 clinical trial, both the participant as well as the individual who's giving the vaccine are blinded. They don't actually know which version you're getting, if you're getting the vaccine or the placebo. And that's to make sure that there's no bias in the trial. The other thing that's important in a phase 3 is that all the data analysis happens retrospectively. So you're not actually looking at the data over time and then potentially biasing yourself as you're going, but you actually have to wait until you've gotten enough people enrolled into the trial and you've had enough symptomatic cases of COVID-19, a predetermined number, and then you go back and you actually look at your data. So that's something that's to keep the bias out of the trial. What we've seen so far is that we've seen some great efficacy numbers out of the SARS-CoV-2 phase 3 candidates that have been tested so far. We've heard numbers like 90-95% efficacy. What does that mean? So in these cases, we're looking at symptomatic COVID-19 cases at the end of the day, and what we're able to say is that 90% of those cases were actually in the placebo cohort as compared to 10% of the cases that was in the vaccine cohort. And that means then that you were much more likely to get the COVID-19 symptomatic cases if you were in the control cohort than if you were in the vaccine cohort, and that's a great outcome. Now we just have to wait and see how well do these vaccines actually work in the real world and what their effectiveness is going to be.